All right. Are we almost all back? I don't know why that happens, and I think it happens more towards the end of the semester than it does in the middle of the semester. But yeah, I don't know why it happens. And before I teach online again with Zoom, I will figure out what the problem is. But because we're 2 days away from being done, we'll just put up with it for right now. Hopefully it doesn't happen again. Any other questions, issues? I'm not frozen anymore. Don't think we have everyone back yet. Okay. All right. Let's jump into tonight. Okay. So troponin and tropomyosin are essentially acting like a switch. You want to see my puppy again? He's right here. You gonna say hi? Louie, look up. Look up. Yeah, hi. Says I'm right here. My mom just woke up, so he's coming out to say hi. He still likes my mom better. He just uses me because I take him on long walks, but he tolerates me most of the time. So troponin, tropomyosin, acts like a switch that is going to determine when the fiber can or cannot contract based on whether calcium is bound to troponin. So the contraction of the muscle is activated by the release of calcium into the sarcoplasm and its binding to troponin. And troponin is going to bind to the calcium and as a result change shape and shift the tropomyosin off the myosin binding sites on actin. Titin is a huge springy protein that runs through the core of the thick filament. It anchors the thick filament to the Z disc and the M line, and it helps stabilize and position the thick filament. It's given its name because of the strength of the protein. Its ability to stretch gives muscle extensibility. And it also provides a recoil that gives muscles elasticity. So the Z disc is made of a protein called alpha-actinin. And the M line is made of accessory proteins, which include myosin C proteins and creatinine kinase, which are connected to titin. So we talked a lot about how the thick filament was attached to the Z disc. However, it's also attached to the M line. Nebulin is another supporting protein that wraps around the entire length of the thin filaments and helps to anchor the thin filaments to the Z discs. I'm listening to a lot of YouTube videos lately, and they're really good because they give me a review of material before I have to teach it. And I'm also trying to put together a series of YouTube videos that I can use in class. And what he was calling the M line something different than I call it, and the Z disc something different than what I call it. And I was watching and listening to it in my car on my way home this afternoon. Don't say anything that I'm watching YouTube videos as I drive. I was really listening more than watching. But I think there's multiple names. But as far as I know, all the textbooks that I have refer to them as a Z disc and the M line or M band. Dystrophin is clinically the most important non-contractile protein of muscle contraction. It works to link the actin to the most outermost myofilaments. It links actin in the most outermost myofilaments to the membrane proteins that are linked to the endomysium. If you remember, the endomysium is around each muscle fiber. And it transfers the forces of muscle contraction to the connective tissues. It helps to transmit tension and help to transmit it, and at the same time, what's the word for it? Reinforce it, support it, strengthen, propagate the tension. Yeah, propagate the tension, that's more the right word. Helps transmit the tension generated by the sarcomeres to the tendons. The gene for this protein is, when it's mutated, it results in Duchenne's muscular dystrophy. Without dystrophin, there's no reinforcing effect of the dystrophin. And this allows the sarcolemma to tear easily. Remember what the sarcolemma is: the plasma membrane of the muscle fiber, which is, for our needs and purposes right now, and as far as I can remember, it's synonymous with the endomysium. And so as a result, muscle fibers rupture easily and die. I know that they're looking into gene therapy for Duchenne's muscular dystrophy. It's probably more common than most muscular diseases are. And I don't know where that is in the process. But the few cases that I've seen of Duchenne's muscular dystrophy are always very sad to me. Before I moved to the East Coast, I lived in Idaho, and I subbed in the school district. And I can say I don't like subbing in the school district. I'm glad I'm not doing it anymore. But I got a lot of experience working with people of all ages. I did everything from pre-K to high school seniors. And I worked with a little boy who was 4 years old, and he'd been diagnosed with Duchenne's muscular dystrophy. And he was so sweet. And I remember just thoroughly, enjoying him. And at the same time, whenever we were out on the playground, he was still walking at that point because he was only 4. And I would, every time we were out on the playground, regardless of whether he should be running or not, I was letting him run. And it was mainly because I knew at some point in his life it was going to be over. And this running would be a memory to him. And so I just remember watching him run and letting him really go wherever he wanted. And I just kind of shadowed and realizing that at some point it would be the end. And usually they die from suffocation. Striations are alternating A bands and I bands. So along the length of an entire muscle fiber, there's about 10,000 sarcomeres banded together. And the sarcomeres are going to extend from one Z disc to the next Z disc. So from one Z disc to the next Z disc is the length of a sarcomere. And they're the functional contractile unit of a myofibril. They attach neighboring sarcomeres and separate one sarcomere from the next, providing anchorage for thick and thin filaments. And these are answers. And if I realized, I can tell you I don't memorize these PowerPoints. I have to look at them before we come back to class. At least I try to every time to remember the order of what's going to come one slide after another. But Max, you asked what A and I stood for, and I had to look it up. Well, it's right here too. And I won't forget now. But A stands for anesiotropic, indicating the way the bands reflect color, polarized light. I stands for isotropic. The A band is where the thick filaments overlap with the thin filaments. And the A band runs the entire length. The band of every A band, the width of every A band runs the entire length of the thick filament. The H zone, which theoretically, it's not as dark. The H zone or the H band is not as dark and is in the middle of the A band. Because this is the place where you don't have the overlap, you just have the thick filaments. You don't have any overlap of thin filaments. Bear with me, the M line is the middle of the H zone. And the I band is just the thin filaments and the Z discs. So this is one way to remember the letter I it comes from, has the word thin has an I in it. The letter H is found in the letter thick. I don't know if that really helps me. I think I still like the I versus the A in that I know that I, because they're thicker, are going to be specifically the myosin, the thick filaments. Because if they were thicker, I would think that they would stain darker. The thin filaments are the I bands. And just the actin by itself. That wasn't a mnemonic I got. I got that mnemonic from another professor. But I don't think I'm going to keep it on there anymore because I don't think I like it. It just confuses me. So just an overview of what we've been talking about. Muscle is a contractile organ, usually attached to the bones by way of tendons, composed of fascicles that are tightly packed parallel muscle fiber bundles that are supplied with nerves and blood vessels and enclosed within a fibrous epimysium that separates it from neighboring tissues. And I don't know if we have it here or not, but the epimysium, I think it says in your textbook that it is an extension of, it's continuous with the tendon. Actually, I think this was where Max brought it up one time, that it's a specialized part of the epimysium. Just remember, it's the most outer layer of the muscle. A bundle of muscle fibers. So a fascicle is a bundle of muscle fibers within the muscle, supplied by nerves and blood vessels and enclosed in a fibrous perimysium that separates it from neighboring fascicles. So it's what's going to wrap around each fascicle. A muscle fiber is a single muscle cell. It's enclosed in a specialized plasma membrane, the sarcolemma, contains densely packed bundles of myofibrils, and the myofibrils contain the myofilaments, actin and myosin. And the endomysium surrounds each muscle fiber. So this would be at the level where the dystrophin is affected, is the muscle fibers attaching to the upper or more superficial endomysium. Myofibril is a bundle of myofilaments. Collectively, they fill most of the cytoplasm. Each is surrounded by a sarcoplasmic reticulum and mitochondria. Remember that we don't generate new muscle fibers. Once you're born, you have essentially all of the muscle fibers you will ever have. They only enlarge. And they enlarge by the addition of new myofibrils. Meaning you increase the number of myofilaments. A sarcomere is a segment of the myofibril that starts from one Z disc to the next Z disc. And each muscle fiber is hundreds of sarcomeres connected end to end. And I messed that up, myofibrils. Myofilaments, actin and myosin overlap and shorten each sarcomere. Any questions about the overall structure of muscle? So stimulus for muscle contraction. The muscle never contracts unless it's stimulated by a nerve. if nerve connections are severed or poisoned, a muscle is paralyzed. Denervation atrophy is when there's a shrinkage of the muscle due to the fact that the nerve fibers have been disconnected. A motor unit is one nerve fiber and all, one nerve, yeah, I said it right. One nerve fiber and all of the muscle fibers innervated by it. So the muscle fibers of one motor unit, and you can have anything anywhere from 3 to 1,000 to several thousand muscle fibers being innervated by one nerve fiber. So the muscle fibers of one motor unit, all of those that are innervated by one neuron, are dispersed throughout the muscle and contract in unison. They follow the all or nothing law in that they're either going to contract fully or they're not going to contract at all. And they are going to produce a weak interaction over a wide area. But with the alternating long-term contractions of individual motor units, you can sustain a long-term contraction. So effective contraction usually requires contraction of several motor units at one time. When a motor neuron fires an action potential, how many skeletal muscle cells in that motor unit contract?
All of them?
All of them, right. So they're all going to contract at one time. So remember, a motor unit is essentially, the size of a motor unit is how many nerve fibers are innervated by one neuron. So the bigger the ratio, is that the right? Yes, bigger the ratio, the larger the motor unit. The smaller the ratio, the smaller the ratio, the larger the motor unit. I don't know if that helps. That's the way I think of it in my head. So a small motor unit is where you have 3 to, for example, is 3 to 6 nerve fibers per neuron. Examples would be those of the eyes, regulate the extrinsic eye muscles. The ones regulating your fingers, where it's not so much strength, it's more control and fine motor skills. Larger motor units are going to produce powerful contractions, where it's more about strength than it is about control. So remember that neuromuscular junctions are essentially synapses. We've talked about synapses before. They are the only difference between this synapse and other synapses that we've talked about, where we had a presynaptic neuron and a postsynaptic neuron. Here, rather than having that, we have a motor neuron that's synapsing on a muscle cell instead of another neuron. We also have neuromuscular junctions that synapse on glands. So the synapse is a point where the nerve fiber is going to meet with its target cell. And when the target cell is a muscle fiber, it's a neuromuscular junction. Each terminal branch of the nerve fiber within the neuromuscular junction forms a separate synapse with the fiber. So if we look, that's what I meant to do. We look right here. This is one synapse, right? So this is one axon terminal synapsing on the skeletal muscle fiber. Each one of these little dots is an individual connection with the muscle fiber. Remember that the axon terminal can make multiple connections with the nerve fiber it's innervating. So the neuromuscular junction consists of a synaptic knob, the swollen end of the nerve fiber, which contains vesicles of acetylcholine. The synaptic cleft, which is the gap between the synaptic knob and the sarcolemma, and the motor end plate, which is the region of the sarcolemma opposite that of the synaptic knob. Louis. Which, and that the synaptic end knobs containing the motor end plate has acetylcholine receptors. The Schwann cells are going to envelope and isolate the neuromuscular junction. So remember, when we talked about the differences between autonomic neurons and somatic motor neurons, autonomic neurons are completely, motor neurons are completely myelinated. So the myelin is going to come down, it's going to cover the end bulb, and it's going to envelope this whole area. We talk about the fact that there's a lot of reinforcing of connections. This is one of those where neuromuscular junctions are reinforced. So acetylcholine gets released from the synaptic vesicle. What ion is involved in that release? I don't remember if we talked, I think we did. What ion is involved in that release?
Calcium.
It is, yeah, good job, good memory. Yes, it's calcium. So calcium, as the oxygen potential comes down the motor end plate, it's going to open voltage gated calcium channels. And the calcium is going to enter the axon terminal and fuse to the vesicles inducing them to fuse with the membrane, resulting in the exocytosis of the neurotransmitter. So acetylcholine gets released from the synaptic vesicles. It diffuses across the cleft and binds to the acetylcholine receptors, which opens sodium channels. The opening of the sodium channels and the voltage change of that opening that results from those openings induces more voltage gated sodium channels to open. So the first ones are ligand gated channels. The second ones are voltage gated channels. We know it's ligand gated because acetylcholine has to bind to it. And then once the sodium starts to enter, the voltage changes of the membrane potential, and that opens more voltage gated sodium channels. I'll tell you just really quickly, I have a professor, my course coordinator at another school thinks that we don't teach enough physiology at in AMP classes. And so I guess I'm trying to prove her wrong by reinforcing this with you, because I think anatomy is important and it's awesome. And that's where I got my start. Was I worked with an anatomist who wrote a lot of textbooks, but the physiology is just as important. And so I hope you're getting it, because when she said that to me, I thought, I make my students learn physiology. I think I do. So then when those voltage gated sodium channels are opened, after the ligand gated sodium channels are opened, that's when the muscle action potential is initiated that moves along the sarcolemma. To keep it from the acetylcholine from sticking around, because we want the neurotransmitter to stick around, but then we want it to go away, because we want to be ready for the next action potential that moves through the neuron. So acetylcholine is then broken down by acetylcholine esterase. And that the components are reabsorbed by the synaptic end bulb. So the resting membrane, the skeletal muscle has a resting membrane potential, and it's more negative than that in the neuron. Remember, the neurons was around -70 millivolts. Skeletal muscles around -90. And there are more anions, and this is because there's more anions on the inside of the membrane than there are on the outside. What's that major anion that's found more on the inside than on the outside? Do you remember? And if it helps, it's not a single atom ion. We have potassium leaving that's positively charged. We have sodium that's trying to enter the cell that's positively charged. What's actually keeping that membrane more, the inside of the cell, definitely more negative?
Is it negatively charged proteins?
It is. Booyah. Yes, it is. It's negatively charged proteins because they're trapped in there. So overall, that's what's actually causing it. We're looking, but when we're looking at immediately along the membrane, that's where the interplay of sodium and potassium is coming about. But holistically, it has to do with the fact that there's negatively charged proteins inside the cell. So there's an excess of sodium in the extracellular fluid, and there's an excess of potassium in the intracellular fluid. The T-tubular membranes contain more chloride channels than neurons do, which makes these cells even more permeable to chloride, in addition to the fact that it also has potassium weak channels like neurons. Is the sodium going to want to enter the cell or exit the cell? Excuse me, bear with me, is the chloride going to enter the cell or exit the cell?
Exit the cell.
You know what? Let me put it this way, it depends. It balances sodium. As Zena can tell you from going through fluid and electrolyte balance in 122 recently, the homeostasis of chloride follows that of sodium. If there's more sodium outside of the cell, chloride will want to be more outside of the cell. But then when sodium enters the cell, chloride wants to join it. However, during an action potential, chloride, the membrane is not as permeable to chloride. That's what makes an action potential so effective. If chlorine could immediately follow it, you'd have a zero charge. But because chlorine can't follow it, that's what makes it more effective. Does that make sense? Maybe. It's always trying to balance it. And when it's not permeable, that makes the sodium more inclined to move back out of the neuron again, and move back out of the cell. So the resting membrane potential overall, because we know that it's really the overall negative charge is coming from those proteins. But when it comes to the charge difference right immediately adjacent to both sides of the membrane, it's maintained by that sodium potassium pump, which is essentially recharging our battery. Recharging that separation and charge, allowing it to continue to do what we want it to do. Any questions about this? Myasthenia, I don't think I'm saying that right. Myasthenia? Mom, how do you say it? Myasthenia? Myasthenia. Okay, so I said it right initially. Myasthenia gravis, an autoimmune disorder, meaning your body is attacking itself. Antibodies, autoantibodies, so your own body's antibodies, are targeting the acetylcholine receptors on skeletal muscles. So the acetylcholine receptors are destroyed by your own immune system. The decrease in the number of acetylcholine receptors results in the slow neurotransmission that inhibits muscle contraction. The result is that you get muscle weakness. What kind of medicines can you think of that would actually work to delay the effects or minimize or help with the effects of this disorder. What are some things that you can think of that would actually, that are probably being bioengineered if they're not already available, that would help treat this disorder and allow people to live a more normal life who have this?
Does it have to do with stem cells possibly, or using like other animal?
I don't know if stem cells would work in this case, and the reason is because your body will start to attack them. What else? What else? We talked about a lot of drugs work on our autonomic nervous system. What were they doing?
You're talking about the ones that were kind of filling in the holes, you know, like the different mental health, like depression type drugs.
Yeah. What were they doing? Do you remember?
They were, it had to do with the receptors. I remember that part. I couldn't remember if they were slowing it down or if they were filling in.
They're slowing it down.
Okay.
So what they're doing, they're not specifically working on the receptors so much as they're working on keeping the neurotransmitter around in the cleft longer. Because even if you start running out of acetylcholine receptors, if the number gets lower, if you can keep the acetylcholine around longer by inhibiting acetylcholine esterase, you can maintain the effects longer. Does that make sense? Bacterial products and drugs can selectively block events at the neuromuscular junction. Botulinum toxin produced by Clostridium botulinum blocks the acetylcholine release from synaptic vesicles of the neuromuscular junction. It can also be found in improperly canned foods where a tiny amount can cause death by paralyzing respiratory muscles. So what it does is it blocks exocytocin of acetylcholine from the neuron. It blocks the fusion of the vesicles with the membrane, resulting in those muscles not getting contracted and relaxing. It's used as a medicine to treat strabismus. I think that's how my mother told me to say it. Strabismus, crossed eyes, blepharospasms, spasms of the vocal cords that interfere with speech, and as a cosmetic treatment to relax muscles that cause facial wrinkles. Any questions so far? So phases of muscle contraction, there's 4 phases. There's going to be an excitation contraction coupling, contraction, and then relaxation. So the excitation is the process in which nerve fibers lead to muscle action potentials. excitation contraction coupling is the events that are linking the excitation action potentials on the sarcolemma to the activation of the myofilaments, the thin and thick filaments, preparing them to contract. Contraction is a step in which the muscle fiber develops tension and may shorten by the overlap of the myofilaments. When the stimulation ends, a muscle fiber relaxes and returns to its resting length. So nerve signal, action potential, comes down the axon, and at the axon terminal, it opens voltage gated calcium channels. Calcium enters and stimulates the release of acetylcholine from the synaptic vesicles into the synaptic cleft. And the acetylcholine then diffuses across the cleft. So this step right here, the second step here, is the one that Botox is working on. It's not preventing calcium from entering. It's not preventing it from binding to its receptors. It's preventing the fusion that's going to allow for exocytosis of the acetylcholine. Two acetylcholine molecules will bind to each receptor and open the channel. And the sodium enters and shifts the resting membrane potential from minus 90 millivolts to positive 75. The potassium exerts and returns, then exits and repolarizing, returning the membrane potential to a minus 90. This quick shift is the end plate potential. This voltage change in the end plate potential opens nearby voltage gated channels, producing a muscle action potential that spreads over the sarcolemma and travels along the T-tubules. I don't know if you remember the PowerPoint that I showed you that we went through on last Wednesday. I've changed it a little bit here. And the reason I've changed it is because it wasn't right. I think right here, yeah, it's the new one. It said the end plate potential, AKA end plate potential. They're not the same thing. An action potential, if you think about it, an end plate potential is essentially a graded potential. Maybe. I'm just trying to make an analogous. When it starts propagating across a membrane and keeps reinitiating itself or self-propagating, that's an action potential. Is that clear what I'm saying? So if it's self-propagating like this, that's an action potential. When it's a quick membrane up-down shift, we more refer to that as being more of a graded potential. So the action potential is going to spread down the T tubules. Remember, those are indentations into the sarcolemma. Spread down the T tubules and open voltage gated ion channels in the T tubules and calcium channels in the sarcoplasmic reticulum. If you remember on your case study, I think it was the second one. It might have been the last one. I think it was the last one. Asks you about calcium channels in the T tubules. They're called ryanodine ryanopyridine receptors. Is this ringing a bell? So what they're doing, so they're opening voltage gated channels, and those channels are initiated by the opening of these ryanodine pyridine receptors that are going to initiate the opening of the channels in the sarcoplasmic reticulum. Calcium leaves the sarcoplasmic reticulum and enters the sarcoplasm. Calcium, once it's released from the sarcoplasmic reticulum, is going to bind to troponin on the thin actin filaments. And the troponin tropomyosin complex is going to change shape. And there's going to be an exposure of that. That's going to result in exposure of the active sites on actin to which myosin binds. And then we get the cross bridging. Oh, you know what? I know what I wanted to show you. Bear with me just for, I'll finish this and then I'm going to show you something that I found the other day. So the myosin heads bind to ATP, and that cocks them into a high energy state. They hydrolyze the ATP, and at that point they can ADP and inorganic phosphate are still bound to them, but the heads are positioned in such a way where they can actually bind to the actin. They then release the ADP, and this shifts the head. Pulling the actin forward. As the myosin heads then bind to another ATP, the myosin can then detach from the actin and the process can be initiated again. So ATP binds to myosin. ATP binds to myosin. The ATP gets hydrolyzed, and this puts the head of the myosin into a high energy state where it can then bind to the actin. It then releases the ADP, and this causes a swiveling of the head, pushing the actin forward towards the M line. Then, in order to be released from the actin, the myosin must bind another ATP. Now, I found a really short video. Let me see if I can find it again. Bear with me just for a second. Oh, I don't want it. Let's see here. Oh, here.
People with ADHD save 10 hours studying with this Chrome extension. But no, by all means, go. Muscle myosin is a dimer with two identical motor heads that act independently.
So here's your thick filament. Here's the myosin chain. Here's the head. Here's the actin. Here's the site on actin where myosin is going to bind.
Each myosin head has a catalytic core and an attached lever arm. A coiled coil rod ties the two heads together and tethers them to the thick filament seen on top. The helical actin filament is shown at the bottom. In the beginning of the movie, the myosin heads contain bound ADP and phosphate and have weak affinity for actin. Once one of the heads docks properly onto an actin subunit, phosphate is released. Phosphate release strengthens the binding of the myosin head to actin and also triggers the force-generating power stroke that moves the actin filament. ADP then dissociates, and ATP binds to the empty nucleotide binding site, causing the myosin.
So it's it's the release of the phosphate, and then that's going to shift it forward. Another way to just think of it is it's going to be the release of that ADP and phosphate, but it can't detach again until another ATP binds.
Head to detach from the actin filaments. On the detached head, ATP is hydrolyzed, which recocks the lever arm back to its pre-stroke state.
So we've talked about the coupling of ATP hydrolysis to other reactions. So that's what's occurring here is you hydrolyze the ATP, and that energy from the breaking of that bond is used to recock the head of the myosin.
Thus, like a spring, the arm stores the energy released by ATP hydrolysis, and the cycle can repeat. The actin filament does not slide back after being released by the motor head, because there are many other myosin molecules also attached to it, holding it under tension. Sliding filament theory is the mechanism by which muscles.
I like that video. It's better. I don't do a really great job. I don't feel like I do a great job explaining that very well, but that's what's going on. Is it clear? So myosin pulls on the actin, causing the thin filaments to slide inward towards the M line. As a result, the Z discs are moving toward each other and pulling on neighboring sarcomeres, which results in the shortening of the myofibril. And thanks to the structural proteins like dystrophin there's a transmission of force throughout the entire muscle, resulting in whole muscle contraction. The lengths of the thick and thin filaments are not changing. It's just the overlap that's changing. So you see a loss of the I bands because that's where there's just, during muscle contraction, you see a loss of the I bands because that's where there's just thin filaments. And as the overlap becomes greater and greater with contraction, the I bands are going to disappear. The A bands don't change. The H zones, there where there's just the thin filaments may change. Excuse me, where there's just the thick filaments may change. And you can see that here. The I band is going to slowly disappear. The H zone is slowly going to disappear. But the A band is going to remain the same. So nerve stimulation and acetylcholine release stops, and acetylcholine esterase is going to break down acetylcholine, and the fragments are going to be reabsorbed into the synaptic knob, halting the stimulation by acetylcholine. Calcium gets pumped back into the sarcoplasmic reticulum via active transport, and the calcium binds to calsequestrin while being stored in the sarcoplasmic reticulum, which actually allows for more calcium to be stored in the sarcoplasmic reticulum. As the calcium is being actively pumped back into the sarcoplasmic reticulum, the concentration of it is falling in the sarcoplasm, allowing the calcium to come off the troponin. The tropomyosin then reblocks the active sites on actin, and the muscle fiber ceases to produce tension, and the fiber returns to its resting length. Overview here. Nerve impulse arrives at the axon terminal of the motor neuron, triggers the release of acetylcholine. Acetylcholine diffuses across the cleft and binds to receptors in the motor end plate, triggering an muscle action potential. Acetylcholine esterase in the cleft will destroy acetylcholine, so another action potential does not arise unless there's more acetylcholine released. The traveling muscle action potential moves along the T-tubule, opening calcium release channels in the sarcoplasmic reticulum, which floods the sarcoplasm with calcium ions. The calcium binds to troponin on the thin filament, exposing the binding site for myosin. Contraction occurs because power strokes using ATP, the myosin heads are going to bind to the actin, swivel, and release, with the thin filaments being pulled towards the center of the sarcomere or the M line. When the muscle relaxes, the calcium release channels in the sarcoplasmic reticulum close, and the calcium active transport pumps use ATP to restore the low levels of calcium in the sarcoplasm. Troponin and tropomyosin then slides back up into position, blocking the myosin actin sites, and the muscle relaxes. Let's talk about one more thing and then we'll take a 5-minute break here. So rigor mortis. Following death, cellular membranes become leaky, allowing calcium to flow into the sarcoplasm from the extracellular fluid and also from the sarcoplasmic reticulum. The increase in the amount of calcium promotes cross bridging because it binds to the troponin, exposing the active sites on actin for myosin. But because the person is dead, ATP is no longer being produced. So the heads are stuck, the myosin heads are stuck on the actin and aren't allowed to detach from the actin. So the loss of the ATP synthesis and other cellular processes that cease following death keep the actin and the myosin filaments linked because of the breakdown of the membranes, allowing calcium in. And the muscle will remain rigid until the myofilaments start to decay. So it's not the ATP that starts contraction, it's the calcium. All right, let's take a few minutes here. Let's come back at 7:30 and we'll finish things up. Or almost finish things up if we don't finish them up. Okay. I can't stop. So much. So much easier. Because it's gonna be outside. Or a really bulky sweater. What do they call formal? You know, I'm wearing a nice Sunday dress because I know her wedding dress is black. I'll wear dark. You gotta love that good inside us. So myself. It's so myself. That's what. Oh, really? Oh, that's funny. No, I'll never forget the cemeteries in Japan because I always thought they were really nice. There's one gravesite right on top of the next, and there's no space. Yeah. Well, no, I told you about the one you found at the old town. That we learned down there. He says, don't you think it's not nice that you're throwing his bones out of the ground? And the guy says, no, he says, when I put the next guy in, I put his bones right back in the top. And I thought, okay, works for me. I'm sure the guy's happy. There was a little right next to Jake's house. There was a little family plot right next to Jake's house there in Stafford. In their big house, the newer house there. I remember walking. Or was it their other dog? I think it was the dog they have. Walking around the little 5, 10 gravestone cemetery. Stafford. Was it Stafford? Didn't they live in Stafford at one point? Or was it Raspberry? I think it was Raspberry. I don't know. All right. It was Raspberry Lake. Yeah, it was Raspberry Lake. All right, let's come back to this. So, length tension relationships. So the length tension relationship of a muscle is a relationship between the length and the force the muscle can produce at that given length. When the sarcomeres are greatly shortened, the amount of tension, you may be able to produce tension, but you're not going to get a significant change in length. Because the myosin filaments are just going to butt up against the Z disc. When there's not a lot, when there's very little overlap or minimal overlap, remember that the thick filaments have to be able to bind onto the actin. And so if there's very minimal overlap, it means that the myosin will not be able to bind well to the actin. So the overall optimistic resting length is about 80 to 120% of the resting length. So turns out that the natural resting length of our skeletal muscles maximizes this ability of the muscle to contract when stimulated. The nervous system is going to maintain a muscle tone to ensure that resting muscles are somewhere near their optimal length. If too stretched before stimulated, there's weak contraction because of the minimal overlap, resulting from minimal cross bridge formation. The optimal resting length is going to produce the greatest force. If you think about it, sprinters don't start a race standing up. They start partially contracted because they want to generate the most force. So in contrast, if a muscle is overly shortened before stimulated, there's weak contraction because the thick filaments just, you may be able to create cross bridge formation, but there's really not much more length that you can shorten. And to help with this, I did remember to post a video about this. I wish I'd remembered the myosin one, but at least I found it fairly fast.
Let's look at a phenomenon known as the length-tension relationship of a sarcomere. When a skeletal muscle fiber contracts, myosin heads attach to actin to form cross bridges. This is followed by the thin filaments sliding over the thick filaments as the heads pull on the actin. This results in sarcomere shortening, creating the tension of the muscle contraction. The cross bridges can only form where thin and thick filaments already overlap. So the length of the sarcomere has a direct influence on the force generated when the sarcomere shortens. This is called the length-tension relationship. The ideal length of a sarcomere to produce maximal tension occurs at 80% to 120% of its resting length. At the resting length of the muscle, the maximum amount of cross bridges are able to form. When the sarcomeres are overly contracted, there is a high degree of overlap between the thick and thin filaments, but further muscular contraction is halted by the budding of the myosin filaments against the Z discs. Tension decreases due to this pause in cross-bridge cycling formation. When we increase the muscle length beyond the optimum point, the actin filaments become pulled away from the myosin filaments. There is little interaction between the filaments. Very few cross bridges are able to form and less tension is produced. When the filaments are pulled too far from one another, they no longer are able to interact and cross bridges fail to form at all, resulting in no tension.
Are there any questions about this length tension relationship? So a myogram is a chart of the timing and strength of muscle contractions. So when the timing between when the action potential is initiated and how long is the muscle contraction, the duration of the muscle contraction. The threshold is the minimum voltage necessary to generate the action potential in the muscle fiber. So just like the thresholds to do for any action potential, we have a threshold here too. So think of it as the minimal pressure necessary to activate a key on your computer. A twitch is a single brief contraction of all the muscle fibers in a motor unit in response to one action potential that reaches threshold. With the subthreshold stimuli, there won't be a contraction because you haven't reached threshold. At the threshold intensity or above, you get a twitch. So looking at the stages of a myogram, there's a latent period, which is the brief delay between the stimulus and when the muscle contracts. This is the time that's required for excitation, contraction coupling, and the tensing of the elastic components of the muscle, generating internal tension. The action potential that was initiated by the motor neuron is going to propagate over the sarcolemma, and the calcium is going to be released from the sarcoplasmic reticulum, and then the calcium is going to bind the troponin. This is all occurring during the latent period. So if we're looking right here, it's occurring right here. And the contraction phase, this is a time when the muscle generates enough external tension to overcome the load, whatever antagonistic weight you have, and cause movement. So the tropomyosin is gonna move, and the myosin binding sites are gonna be exposed on the actin. The cross-bridge cycling is going to occur, and the sarcomeres are going to shorten. The relaxation phase is the time when the tension declines back to baseline. So the sarcoplasmic reticulum is going to reabsorb all the calcium via active transport. Myosin heads are going to detach from the actin, and the tension is going to decrease. And the tropomyosin, because the calcium is no longer bound to the troponin, is going to cover the actin binding sites. And this phase overall takes longer than the contraction phase. there is a refractory period, which is the time in which the muscle fiber contracts. Just like we saw with the sodium absolute refractory period. When the muscle fiber is already contracting, you can't initiate it to contract more. Do you remember that absolute refractory period? When the sodium gates are open, you can't open them more, they're already all the way open. It's the same thing here. When the muscle's contracting, you can't make it contract more. It's already doing its maximum. So even if the same voltage is delivered, different stimuli cause twitches varying in strength because the muscle starting length is going to influence tension. Remember the length tension relationship. Muscle fatigue occurs after continual use. And warmer muscles are going to work more quickly than colder muscles. You're going to get better muscle use after you've been working out for a little bit. Another example, and it might have to do with warmer muscles. We talked about, I think we talked about, as I'm going through muscle in 2 different classes, we talked about the glycolytic versus oxidative energy levels. Once you are moving into oxidative muscle use, muscles tend to work better. I can tell you, I work out about once a week, and I work out for about 1.5 hours, and it's usually my self-care time because I just realize that emotionally I do better throughout the week if I can get that in. But for the first maybe 10 to 15 to 20 minutes, I feel like I'm hitting a wall. And after that, I can do great. Because it takes a while, like we talked about towards the very end of lab last week, it takes a while for your respiratory and your cardio system to get up to speed. That has to do with the warmer muscles. Also, the muscle cell's hydration level is going to influence cross bridge formation. Structure function relationship. And increasing the frequency of stimulus delivery is going to increase tension output. If the action potentials are coming fairly quickly, you're going to get better tension output. Questions? So muscles must contract with varying strengths for different tasks. Stimulating nerves with higher voltages will produce stronger contractions because different muscles have different thresholds. Actually, let me put it this way. Different motor units have different thresholds. Higher voltages will excite. Hey, buddy, Louie. You keeping us safe, buddy? You keeping us safe? Yeah. They don't realize what a softie you are, but we won't tell them either. So higher voltages are going to excite nerve fibers with higher thresholds that require a higher threshold stimuli, stimulating more motor units to contract at one time. The strength of the contraction, therefore, depends on the strength of the stimulus. At a subthreshold stimuli, there's no observable contraction. A muscle is going to increase its response from the threshold up to a maximal stimulus. And any stimulus above that maximal will elicit the same response. Low frequency stimuli produce identical twitches. Higher frequency stimuli, where the action potentials are coming in at a higher frequency, are going to produce temporal wave summation, just like we saw with graded potentials. Each new twitch will ride on the previous one, generating higher tension. And there will be only a partial relaxation between the stimuli, resulting in a fluttering or incomplete tetanus. All of this incomplete tetanus is going to result from the fact that additional calcium is being released from the sarcoplasmic reticulum before all of the calcium is pumped back into the sarcoplasmic reticulum, allowing the muscle to only relax partially. It takes time to pump back all the calcium again. And so if you have another twitch, another action potential that results in a threshold stimuli that initiates another twitch before the first twitch has ended, you're going to get summation. So stimulation at a higher rate results in a sustained muscle contraction that permits partial relaxation between the stimuli. This is called unfused tetanus or incomplete tetanus. So unfused and incomplete tetanus are the same thing. Unnaturally high stimuli frequencies, like in lab experiments, can cause a steady contraction called complete fused tetanus that you don't observe in natural environments. So the complete fused tetanus is not something that you normally observe. You're not going to experience it as a living person. He likes me best right now. What are you doing to him? No wonder he likes me best. My big dog that normally likes my mother best is hiding behind me. because he likes me best when she's getting ready to mess with his ear. He came to us with a raging yeast infection in his one ear. So she's now got a vet appointment for tomorrow, but we've been keeping it reasonably under check. He says, I'm not coming near you.
Come on, get up,
He knows
Get up.
better.
get up.
He's
I'll
a
come
smart
to you
boy. He
here.
knows better.
I can work under the table.
Mom, if you make him yike while I'm in class,
He
you're in
doesn't
trouble.
know.
He's cowered more under the table than he was before. All right. So strength of muscle contractions depends on the size and number of motor units. Larger motor units are going to generate more tension due to the larger number of muscle fibers. As the number of motor units are activated, the muscle is going to generate more and more tension to lift heavier loads. All of the fibers in one motor unit contract in unison. We already knew that, all at once. Muscle fibers of a motor unit are dispersed throughout the muscle and not clustered together. So motor unit recruitment is the process by which the number of active motor units increases. The weakest motor units with the lowest thresholds are recruited first, followed by stronger motor units. The recruitment of additional motor units is going to result in more contraction or motor unit summation. With increased stimulation, you get more contraction because more motor units are having their thresholds reached. And the motor units are going to contract alternately. Remember how we like to alternate them so that we can maintain set contractions for longer periods. Once you've recruited enough motor units, you don't need any more. Questions? So what is in the brain? What is actually inducing these motor units to be recruited?
Neurotransmitters.
Neurotransmitters, but how are they being sent? What's specifically in the brain that's inducing this? What structures? It's the cerebellum and the cerebral cortex, providing that feedback. How much weight, you can push a car, but what if that you go to start pushing the car and you realize it's full of bricks inside? A lot heavier than you thought it was. Not that I've ever been able to push a car by myself. Have I? No, I haven't. But then you realize it's a lot heavier than you thought it was. So your cerebellum induces the recruitment of more motor units because it's the one who's giving the feedback. What if you go to think you're going to push the car like I would? You think you're going to push the car. You're not expecting to be able to push it very far, and you realize it has no engine. And so all of a sudden it's a lot lighter than you thought it was. So the cerebellum is lowering how much motor recruitment and is involved in controlling how much motor unit recruitment you need. Does that make sense? Mom, if you make him yank under there. If a muscle contains a limited number of motor units, then how do we get stronger as we grow up or lift weights? You guys, if you can hear my dog, my mother is messing with his ear under the table. Strength conditioning doesn't increase the number of motor units. It doesn't increase the number of muscle fibers. It's going to result because of the addition of more thin and thick filaments or myofilaments, which are needed to generate more force. Satellite cells are going to divide and fuse with individual muscle fibers to assist in repairing and damaging fibers and growth as you work out your muscles. Any questions? We have discussed, okay, so tension is dependent on the rate at which the nerve impulses arrive. The amount of stretch before contraction is that length tension relationship. And the size of the motor unit. Well, how do muscles meet their energy demands during rest and exercise? You guys, where is ATP used in muscle contraction? There's 3 major places. Where is it used? Where is it used?
In the myofilaments.
It's used in the myofilaments, so the myosin is going to use it to be released from the actin and then to move back into a high energy state again. Where else is it used? Where else is it used?
In the gated channels.
Say it one more time.
The gated channels in the sarcolemma, whatever the membrane is.
You're okay. Welcome to my reality. The sodium potassium pump is one of them. Where's the other one? So the sodium potassium pump, because we have to be able to regenerate that membrane potential every time. Where else? Where's one more? Somebody besides Max. Where's one more?
It's used in the power stroke.
Yep, that was the myofilaments, the myosin. Where else is it used? Let me give you a clue. Muscle relaxation. Where is it used there? He's done with you. You're just lucky he's your favorite, is what it is. So where else is it used, guys? So we have to actively transport the calcium back into the sarcoplasmic reticulum. And so that's where else it's used. So it's used in the myosin, where being able to release the actin and be ready for another round of cross bridge formation. It's also used for recharging the sodium potassium concentration gradients, electrochemical gradients. And it's also used for actively transporting the calcium back into the sarcoplasmic reticulum. So there's three major places where ATP is used. So hydrolysis of the ATPase on the myosin reorients and energizes its head. The binding of the myosin activates the detachment and powers the calcium active transport pumps. This says that there's three. You guys, there's only two. No, sorry. This says that there's like four. There's only three. So muscle contraction demands enormous amounts of ATP. A contracting muscle can use approximately 2 million molecules of ATP every second. Without the constant supply of ATP, muscle contraction ceases. There's two major ways that we get ATP. We can either generate it aerobically, which produces far more ATP and does not generate lactic acid. Or we can produce it anaerobically, where the cells are going to produce ATP in the absence of oxygen, yielding very little ATP and generating lactic acid. We use both. In the initial part of, in the beginning parts of exercising, or with quick exercises like pole vaulting or cartwheel or doing a handstand. All these things I've never been able to do. But all of lifting a weight, I can do that. Lifting a weight. All or activities that are requiring just high, short, intense exercise. So the oxygen is briefly supplied by myoglobin, a derivative of hemoglobin that's stored within the muscle. But this is rapidly depleted because remember, we're only using 2 million ATP per second. Afterwards, two enzyme systems are going to control phosphate transfers to ADP to produce ATP. Myokinase, remember that kinases are enzymes that add phosphates to things. So a myokinase is going to transfer a phosphate from one ADP molecule, one adenosine diphosphate, to another ADP molecule, converting one of those ADP to ATP. So ADP, myokinase takes a phosphate from ADP, converting that ADP to AMP, adenosine monophosphate, and transfers that inorganic phosphate that came from this ADP to another ADP to produce ATP. creatinine phosphate, excuse me, not creatinine phosphate. Well, yeah, kind of creatinine phosphate, but creatinine kinase is going to produce creatinine phosphate. By transferring a phosphate group from creatinine phosphate to an ADP to produce ATP. Both of these phosphate transfers are going to be are part of the phosphagen system that provide nearly all energy needed for short bursts of activity. So initially, before the cardio system and the respiratory system are up to speed, we have to get the ATP somewhere. And this is how we're getting it. And so no oxygen is needed here. We're just transferring the phosphate groups from one thing to another after we've replenished the myoglobin. As muscle activity continues, the supply of creatinine phosphate is depleted. And the muscles will start to obtain glucose from the blood and their own glycogen stores. Remember, glycogen is a stored glucose. In the absence of oxygen, glycolysis can generate about 2 ATP for every glucose molecule consumed. But lactic acid will be produced unless it's aerobic, which is not the glycol, and not right now. In the absence of oxygen, it's going to be completely anaerobic and lactic acid will be produced. You only get 2 ATP for every glucose molecule. After about 40 seconds, or it definitely feels longer than 40 seconds for me, and it may be that my muscles have to warm up. But after about 40 seconds, the respiratory and cardiovascular systems start to deliver oxygen fast enough for aerobic respiration to meet most of the muscle's ATP demands. And at that point, you can get 36 to 38 ATP per glucose molecule. For 30 minutes, for the first 30 minutes, energy is going to be coming equally from glucose and fatty acids. And after 30 minutes, depletion of glucose causes fatty acids to become the major source. Becoming a significant fuel. Questions? We, famous last words, but I'm looking at where we are right now, and we're a lot further ahead than I thought we would be. Let me finish the PowerPoint because I need to finish this, but then let's take a minute and talk about these processes right here. Because I feel like it's a deficit for you guys to not understand this. And you're not going to be quizzed on it, but I think you should understand it better than we haven't covered it. And I think Zoom can tell you that I'm not getting all the chapters done for 122. And we would probably cover this in the almost one of the last chapters, but let's cover it a little bit tonight. So muscle fatigue is due to the inadequate release of calcium from the sarcoplasmic reticulum. You start running out of calcium. Depletion of creatinine phosphate, oxygen and nutrients, the buildup of lactic acid and ADP, and the insufficient release of acetylcholine at the neuromuscular junction. As you keep having one action potential after another fired at your sarcoplasmic reticulum, the sarcoplasmic reticulum stops releasing as much calcium every time. That's where that inadequate release is coming from. So why do you breathe so heavily for a period after stopping exercise? Excess post-exercise oxygen consumption. This time of heavy breathing is going to work to meet a metabolic demand, the oxygen debt, which is a difference between the elevated rate of oxygen consumption following exercise and the usual resting rate. Extra oxygen is used to pay back or restore metabolic conditions to the resting state. This excess post-exercise oxygen consumption is going to aerobically replenish the ATP, replace the oxygen reserves on the myoglobin, provide oxygen to the liver that's busy trying to get rid of the lactic acid, and provide oxygen to cells that have metabolic elevated metabolic rates right after exercise. So this time of heavy breathing can be 6 times the normal consumption and last up to an hour. To delay the onset of muscle fatigue, not all motor units in a muscle will contract at the same time. Some will remain relaxed while others are contracting. If the contraction continues, greater force is needed and more motor units will be recruited. True or false?
True.
It's true. When you are at rest, your muscles are still contracting. Skeletal muscles are always going to exhibit a small amount of tension to help stabilize the position of bones and joints. Muscle tone is established by neurons in the brain and spinal cord that excite the muscle's motor neurons, like your substantia nigra and basal ganglia. Small groups of motor units are alternately being active and inactive in a constant shifting pattern to keep the muscle firm. Also, skeletal muscle fibers are going to vary in their amounts of myoglobin. When you eat turkey during Thanksgiving or fried chicken or something to that effect, dark versus white meats has to do with the amount of myoglobin present. Red muscle fibers tend to have more myoglobin, contain more mitochondria, and are supplied by more capillaries to help maintain the oxygen levels. White muscle fibers have a lower myoglobin content and appear lighter. The diameter of the fiber, so in addition to how much myoglobin they contain, the diameter of the muscle fibers, because remember that's how they're enlarging, that's how hypertrophy is occurring. The contraction of the velocity or contraction velocity, and the method of ATP production, how much myoglobin is there, will determine just what the varying levels of ATP production, developing varying processes of ATP production are occurring at any given moment, and resistance to fatigue. Muscle fibers can be classified into 3 types. Whether they're oxidative, meaning they're using aerobic respiration, whether they're in between, they're being used at the beginning, end of oxidative and the beginning of glycolytic, or whether they're glycolytic. The slow and fast actually has to do with how quickly the ATP gets broken down on the myosin heads. Fast fibers tend to break down the ATP faster. Slow fibers don't break down the ATP as fast. So fast glycolytic fibers are used for shorter sprints because it's anaerobic and the ATP is hydrolyzed fast. Intermediate fibers tend to be somewhere in the middle, rather than really long durations, medium durations. And slow oxidative, meaning that the ATP may not get hydrolyzed as fast, but there's a lot of ATP present because you're using oxidative energy sources. So the slow or fast depends on how rapidly the ATPase in their myosin heads is hydrolyzing the ATP. In marathon runners, the slow oxidative in the quadriceps femoris muscles is about 82%. In marathon runners, fast glycolytic is about 18%. In swimmers, it's about 74% fast oxidative and 26% for slow oxidative, and fast glycolytic is 26%. Average males about 45 and 55. In sprinters and jumpers, where you're going to be pole vaulting or really, really, really fast sprints, fast glycolytic tends to be about 63% of the fibers and 37% slow oxidated. Questions? This change in the percentage is just based on how are you using those muscles. Marathon and swimmers tend to go for longer periods. Sprinters and jumpers need it fast. Questions? So last part here, smooth muscles contain thin and thick filaments that aren't arranged in sarcomeres. That's why we don't have striations. The intermediate filaments or myofilaments. Oh, actually, bear with me. I'm saying this wrong. Yes, I'm saying it wrong. We don't have Z discs, but we have dense bodies that act like Z disc, working to hold on the myosin and the actin. Right here, here's your dense body, here's your next one. Here's your thin filaments, here's your thick filaments. So rather than having Z discs, we've got dense bodies that act like Z discs, pulling on the thin filaments and the thick filaments. So the action spread potentials, rather than being spread along the sarcolemma, they're kind of spread along the sarcolemma, just not to the same extent, are going to be spread through the fibers via gap junctions. And the fibers are normally stimulated by the autonomic nervous system, not the somatic motor nervous system. So these are involuntarily controlled rather than voluntarily controlled. And they can be autorhythmic. Each smooth muscle is going to act like one giant sarcomere. The way that I think about the smooth muscle is that it's very similar, except for you don't have demarcated striations, and the dense bodies are acting like Z discs, and they can start pulling the whole thing up. So smooth muscle is found in the walls of arteries and veins. It's found in hollow organs like the gastrointestinal tract and in the airways of the lungs of bronchioles. It's also found in muscles that attach the hair follicles like the erector pili muscles and the integumentary system, and adjust the pupil diameter. So your oculomotor neuron that's going to adjust the iris and adjust the focus of the lens with the ciliary bodies. Smooth muscle contracts slower and lasts longer than skeletal and cardiac muscle contractions. And it can shorten and stretch to greater extents. And it lacks striations and sarcomeres. And you guys, that is the official end. You have questions for me? I think you're on mute, Zena, if you're talking to me. I don't know if she is.
I'm here.
Question?
No.
Okay.
Sorry.
Let me just show you something just for a moment. And I'm gonna bear with me because I'm gonna have to switch. I'm gonna switch school sites to do this. Oh, I didn't mean to go all the way down here. Let's see here. Let's get this one. So what I want to show you, and I hope this works, let's see how well it works. So this is the 4 phases of how you actually generate ATP. Glycolysis is going to break down glucose into a molecule called pyruvate. This is anaerobically, where one glucose molecule is going to produce 2 pyruvate, and the pyruvate will get fed into the preparatory reaction. But this is going to produce 2 ATP and 2 of these NADH molecules, which are electron transport molecules. The pyruvate gets converted into acetyl-CoA, a 2-carbon intermediate. And in addition to being converted into acetyl-CoA, it's going to produce some more NADH, which is essentially something that's been reduced, and then it will get fed into this process, which will oxidize it and produce a lot of ATP. This process right here, glycolysis, always occurs anaerobically, and it's only going to produce 2 ATP per glucose molecule. The preparatory reaction isn't going to produce any ATP, but it is going to produce NADH, which can be fed into this process down here. The acetyl-CoA is really great, and it's also what fatty acids are broken down into, because it can be fed into this citric acid cycle or the Krebs cycle or the TCA cycle. They're all the same thing. Krebs cycle, citric acid cycle, or TCA cycle. They get fed, the acetyl-CoA gets fed into this cycle in the mitochondria. Remember, that's your ATP powerhouse. And you produce NADH and FADH2 and 2 more ATP, 1 for every acetyl-CoA. And then the NADH, all of the reduced coenzymes get fed into the electron transport chain, which is going to produce another 32 to 34 ATP. So you get, for every glucose molecule produced, anaerobically, you get 2 ATP. If we can make energy aerobically, we can produce NADH here, which will get fed into this process. We get NADH and FADH2, which will get fed into this process, and 2 more ATP here. With all of these NADH and FADH2 molecules that get fed into this electron transport chain, will then make 32 to 30, excuse me, altogether will make 36 to 38 ATP. So this is the goal, is to be able to get these processes going. So initially your muscles are working just on glycolysis. They're only doing this. But if we can get enough oxygen circulating and our respiratory and our cardiovascular systems get up to gear, then we start producing all this. And let me show you one more thing. I hope this is making sense. Feel free to interrupt me here. So one other thing right here. So this is a metabolite pool. What were the major biomolecules? Do you remember? What were the major biomolecules? If it helps you, 3 of them are listed right here.
Are they protein?
Proteins and what else?
I had the second one. I couldn't think of it now.
What is it? Can you see it here?
It's right there.
Carbohydrates and then fats. So proteins can be broken down or they can be produced. If you break them down into amino acids and then you remove the nitrogen groups, you can use them as intermediates in the reactions of glycolysis or the citric acid cycle. Carbohydrates can be broken down into glucose and then fed into glycolysis to produce pyruvate that can be then made into acetyl-CoA. Fats can be broken down into glycerol and fatty acids. Glycerol can be made into intermediates that can be fed into glycolysis. Or fatty acids in each 2-carbon fatty acid of a fatty acid chain, you remember those hydrocarbons, can be made into acetyl-CoA and be used in the citric acid cycle. What this is essentially showing is how the balance between anabolic and catabolic reactions. This is also why, if you're familiar, and some very few of you are old enough, probably Sherry is probably, I think she's one of the older people here with us. I don't want to know how old she is, Justin, if she's not here anymore, but
I would never divulge a
do
woman.
not tell that. That's not more information than we want. We should
I
probably.
don't care. You know that, Justin. I don't care.
Sherry, how old are you?
62.
Oh, wow. You don't look 62.
Just-
Okay.
Just turned it.
Wow. So Sherry, do you remember Karen Carpenter?
I sure do.
Do you know how she died?
Oh, crap. No, I don't.
Anorexia.
Oh, look
Well,
at this.
she didn't die technically of anorexia. She technically died of a myocardial infarction,
Ah.
which was probably the result of anorexia. Why was it the result of anorexia?
Probably because she had gone through all her energy stores and then her heart didn't have have the proper energy to take care of itself. So the tissue started to die.
Well, or you're right. The proteins of the heart get broken, the muscle gets broken down to produce intermediates for these processes. I told you about my cat going from 9.5 pounds to 6.5 pounds and losing 1/4 pound within a day. That's what that is. But with regards to muscle, this is what we were talking about. This is what, when we're doing glycolytic and we don't have our oxygen stores, we're just producing 2 ATP per glucose molecule. But once our cardiovascular and our respiratory systems are where they need to be, we will generate 36 to 38 ATP per glucose molecule. But it takes a few seconds or 40 seconds to get to this point. Does this clarify a little bit about what was going on the other night? Because I felt like I didn't like leaving you kind of stranded with what that was. So bear with me here. Oh, I don't have it down here. Okay. Really quickly. Looking at this. Oh, not there, too far down. This is this. So the Krebs cycle, or the electron transport chain, is this right here. Or
Alan hates the Krebs cycle. He has PTSD from it. He says it's the one thing that he has never been able to actually learn. And Michael says that he learned it, but now absolutely does not remember a thing about it.
this is all you need to remember. Oh, that's funny. I can tell you, I don't remember a lot of the intermediates. I go through it every semester right now, and I don't remember all of the intermediates. It helps that I just went over it about 2 weeks ago. But so this right here, glycolysis to the preparatory reaction. Acetyl-CoA is moving into the citric acid cycle. So looking at this right here, fatty acids are being produced into acetyl-CoA, which is feeding into the Krebs cycle. Amino acids are being broken down from proteins and being used as intermediates in the Krebs cycle, or glycolysis. Pyruvate or pyruvic acid, remember that when it ends in ATE, it's an acid. So pyruvate or pyruvic acid is the same thing. Being fed into the Krebs cycle. Or oxygen coming from hemoglobin once we get aerobically where we need to be. And I have, oh, you know what? If you'll give me 2 more minutes, I know we're beyond time. You don't have to stay. One more video. It's not a very long video. I will, I'll post this as a link. So just talking about cellular respiration, and it's a quick 4-minute video.
The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a
I
snack.
think we're at the end of the video
and
here.
ends with capturing.
No, it's at the beginning. Is it? Are you right? You might be. Okay, you're right. You win the prize. Thank you.
How do you turn? The goal of
Oh,
cellular
stop that.
respiration is to transfer the energy from the
No.
food that we eat
No, no,
daily into ATP that our bodies can use. This process starts with the eating of a snack. Recognize and use as energy. The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients like the carbohydrate glucose will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a 4-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of 2 distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, 2 ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the 2 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction
So
that
that-
involves the
So that NADH was made, and then the ATP was made. And what she was saying initially is you have to put 2 ATP into the process, and then you'll generate 4 ATP. So your net yield will be 2 ATP.
Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound.
So this is going to be that preparatory reaction. You're going to produce 2 NADH and 2 acetyl-CoA, but you can only do this when there's oxygen present.
Called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide.
If you remember at the very beginning, when in chapter 2, we talked about oxidation reduction reactions, how you add electrons to reduce something, and when it's an organic molecule, it also adds hydrogen. So that's these NAD is being reduced to NADH.
The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic.
So this is where carbon dioxide is actually formed. You know, we know that we breathe out carbon dioxide. This is where it's being formed. This is why you produce it.
Respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain.
So the electron carriers, meaning NADH and FADH2, are all fed into this to build up a hydrogen ion gradient out here that will then flow through here and make that ATP.
From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
Okay, I think that's the end. I don't know why it kept going after that because I didn't pause it. Does that make a little bit more sense? Maybe a little bit. Maybe it just made it a lot harder.
Is that video posted?
I will post it for you.
Thank you.
I didn't think about it until just a couple minutes ago. Bear with me. I'm gonna look at one more thing really quick. Just looking at your textbook. Okay, never mind. I'll post it for you. I thought your book would have something about it, and I don't see anything, so I'll post it for you. You have any questions I can answer between now and Wednesday? Guys, have a good night, all right?
Hi, thank you.
Thank you.
You.
Thank you. Wednesday. Have a good night. Thank you for understanding.
No, I understand. How
They
did he
won.
win the game?
They won. Yes, they won.
Awesome. Awesome.
Have a good night. Thank you.
You too.
I was trying to do Smart Book 13. It
Mhm.
won't let me. It's saying only review mode. Isn't that open?
Smart Book 13. No, but I'll open it for you right now.
Oh, okay, that's why. Okay, that makes sense.
No, 1 second there. Um, oh, I didn't mean to do that. Okay, manage extensions. I don't know why you're on here twice, but you are.
I don't know.
You think you could have it done by this Friday?
Um,
What about this Saturday?
yeah, that sounds good.
Okay. Okay, you should be able to do it.
Okay. And the other one was that I noticed, which one was it? 11. I did it and then it didn't give me the whole point before.
What was it, the smart book or was it?
Yeah, it was the Smart Book chapter 11. And then I read it, but then I didn't submit it. I realized that, and then
Give me
I
one
see.
second. Give me one
Okay.
second. Let me look and see what's going on. Smart book chapter 11.
Yeah.
Okay. It looks like you did it all.
For
So,
whatever reason, it only gave me like 2/10.
okay, well, I'll fix that right now.
Okay. I did it the first time and then didn't record it. I did it the second time again. I should have recorded it, but I don't know. Something wasn't right there.
Um, I gave you 10. Are we talking about the Smart Book 11?
Yeah, it was
gave
showing
you
me
10.
on my grades 2/10 and I was confused. Okay, all right.
Yeah, it says 10 out of 10.
It does? Okay, okay. Maybe it didn't update it or something at that time, maybe.
Possible.
Okay, sounds good. All right, thank you so much.
Of course.
Have a good night.
You too.
Thanks.
Jennifer, Kara, are you here?
I just wanted
I'm
to,
here.
oh, sorry. With the extra credit,
Yes.
like some of them I have like 0.87 or 97 or something. Is
Uh-huh.
that like the actual, like, how does that work?
What do you mean?
Like, as far as the number, like, is it that exact number will go towards whatever
Yes.
I'm missing? Okay. I just wanted to check because I was going to work. I was calculating like what points I was missing to make sure I
Okay.
do enough extra credit to cover them.
Right.
Time, but I'm trying to get them done.
Right.
So I just wanted to check because I was like, it's weird and I don't know how I got some wrong, but I did. So
Okay.
I just wanted to check. And then you already opened 14. That was my other question because I had not put, I wrote a note with all my due dates. So other than that, I'm good to
I
go.
think I did. Let me look. Bear with
Even
me.
if it's Wednesday, it's okay. I just,
Well, let me
was-
do it right now, because if I do it right now, I'll actually remember.
okay. There was just so many things.
No, I didn't. What was it? The Smart Book and chapter 14? Was it the Smart Book and the quiz?
I there was nothing posted. It was just the like the slides.
Okay, so that's because they're closed. Do you need me to open them both?
No, you can wait till Wednesday, honestly. If you have it set to open, I can work on them this weekend.
Oh, you're referring to Wiley. You're
Yes.
referring to Wiley. All right.
Sorry.
I thought, I don't remember her asking about this.
Yeah, no, it's the lab.
No, yeah, it should be open. Can you open it now?
I didn't try. I know you opened it earlier because you
Can you
said
do
in like,
me a favor and try?
yeah, let me go.
Let's just see what happens. Because if I told you I opened it, I want to make sure it actually opens for you.
I have, yes, because now there's the lab exercise and questions in the actual section. Before it wasn't there. There was nothing else.
Okay, awesome. All right.
I'm just trying to open, but it's spinning. Hang tight. Open assignment. Okay. Yes.
Awesome. Okay.
That works. And that was all I needed. Everything else, unless you add stuff, I have written down, and I only have like 4 more things to do. So
Okay.
okay.
I haven't. That's the last thing will be the lab quest.
Yeah. The lab quest 5.
Yes.
I have that.
Think that's open. I think it's open.
You
Okay.
opened it the couple, I think, last week because I have that too as 5, 6.
Okay.
So, and then 12 and 13 questions, and then I just have the exams, I believe. Oh no, I do have one more case study that's due on May 6th. Oh, I wanted to ask you about the case studies.
All right. No.
So I know I asked you about the first one because I know, like I said, I didn't know if I did stuff wrong versus not, and I know you weren't grading them.
You know what? Let me look at yours really quick, and I'll tell you.
Because I wasn't sure, like, if they have to be typed, if it has to be like full.
They need to be legible.
Okay.
Let me look at.
I started typing case study 2 when I submitted it yesterday, and I have a Chrome, so it doesn't have Word as an app.
Okay.
And it like reconverted all the numbers. So I took all of my answers and then hand wrote them and then submitted it that way because I was like, she's going to be so confused because it literally reconfigured all the numbers. Enter to the next one when I would answer it.
I think I can read what you've got.
Okay, if not, I can type it. I just didn't want to get points stacked off because I will do it however you need to do it. I just wasn't clear on like what your
Actually, you did a pretty good job in all honesty. Because
Okay.
one thing that I noticed a lot of people last semester when I did this first case study, they wouldn't include a legend.
They would what?
They didn't include a legend.
Oh,
And you did a great job, actually. I'm just looking at the first page,
okay. Of the first case study or the second?
the first case study.
Okay, so I think my writing was a little more scribblier, but I did write clearer for the second one.
Oh, well, it's legend. I can read
Okay.
it.
Okay.
Give me a second. I'll look at the second one. Just a sec.
And did you like want full sentence answers, or is it okay just to answer it how I answered it with just like the creating?
For which one?
For any of them, like in the question, besides like if it was a full detailed sentence.
No,
like,
if the answer's there, the answer's there.
okay. I just want to check. I was like, I don't want to get points off. I'll redo it because I know we have until whatever date to submit it.
Yeah, it looks good.
Okay. I just want to check. I'm gonna do the third one, but I was like, before I do it, I want to ask her.
Nope, you're good. You got
Okay.
it.
Sounds good. Thank you so much.
You're welcome. Have a good night, okay?
Thank you.
Bye-bye.